Okay, welcome to Human Biology at Bethel 1.0 online, or Online 1.0. I'm trying to use an old GoPro, and I've got a remote mic. I hope this sound is adequate for you. I have tried to download or to put as many images on Moodle that I'll be using in these lectures, and so it's nice to have these online because you can pause them at any time, but you may want to open up some of those images so they're readily at hand when we get to them. I'm also trying to use this whiteboard without the screen because I don't have to roll the screen up and down to write something on it. I know there's that seam through the middle, so if that's a problem, let me know, and maybe I'll just pull the screen up and down, but for now we'll try to use it like this. We left off in another life before break with the circulatory system. We were talking about elements of blood. We talked about plasma in blood and the types of proteins that were in it. And we left off with what were called formed elements, the cell types, cell parts that are in blood. And we were dealing with red blood cells. And this image is from your textbook, and the point of it was to make the point that all blood comes from blood-forming tissues in the long bones, basically bone marrow, and the scapula, vertebrae, things of that sort. So in this case, this young lady's femur is being highlighted. There are stem cells that come from blood-forming tissues in that bone. And those stem cells then give rise to all the types of formed elements, cell and cell parts, that are in blood. And so here we see red blood cells, we see all 5 of the white blood cell types, and we even see the platelets. All of those come from this one parent cell, this one stem cell. So we started with red blood cells. Obviously, if you remember from lab, that was the most common type of blood cell, by far mostly what you saw when you looked at your blood slides. We talked about the fact that they're made of or contain proteins. Actually, what I wanted to start with was, if you remember this little ruler from lab, just to give you some scale, on the smallest portion, the centimeter millimeter side, the smallest division on this ruler is a millimeter. And so if you can picture a tiny little cube, 1 cubic millimeter on each of 3 sides, if you can picture a tiny little cube of that size, you could fit roughly, on average, about 5 million red blood cells into that little 1 cubic millimeter container, if you will. So they are very small. There's 5 million in a cubic millimeter. So there's a lot of them in there. And so if we take one of those blood cells out and look at it, and I'll put a picture up here, just so you have something to visualize. Okay, so here are red blood cells. Of course, this is the white blood cell. If you take any of these red blood cells, okay, any of these red blood cells here, again, 5 million per cubic millimeter. If you were to open it up and look inside of it, we would see proteins. There is the molecule hemoglobin. It is a globin protein. There's a picture in your textbook that attempts to illustrate what that looks like. Good picture. Okay, so right there. This is what the artist's rendition of a globin protein looks like. There are 4 polypeptides that make it up. Each polypeptide contains a heme group. A heme group is just a molecule with carbons and nitrogens in it. Nothing particularly you need to remember about it. But you do need to remember that it is the heme group that carries iron, and the iron that attracts and holds the oxygen. Okay? So the heme group has iron in it. Maybe you've heard of iron poor blood. Without enough iron, you don't have a heme group that's functioning like it should, and you don't have the ability to carry oxygen that you should. Okay, so here's your standard hemoglobin molecule. And in that one blood cell right here, if you open that blood cell up, you would find about 250 million of these hemoglobin molecules inside each of those red blood cells. And again, if you remember, there are 5 million red blood cells in 1 tiny cubic millimeter. So gives you an idea of the scale of things. So that hemoglobin molecule made of 4 polypeptides. Again, these are made as amino acids are linked together. You get a polypeptide, you link the polypeptides together, you get the protein. Each of the polypeptides has a heme group. Okay, the heme group is what has iron and is what holds an oxygen molecule. 250 million hemoglobin molecules per red blood cell. What that means is each of those red blood cells has the capacity to carry and hold and release to provide to the tissues 1 billion oxygen molecules. Say it again. Each of these red blood cells can and usually does hold 1 billion, that's billion with a B, oxygen molecules. Just think of your oxygen carrying capacity. If you remember when we had the pulse oximeters, those little things we put on your finger in lab, they were showing your percent oxygen saturation. We didn't talk too much about that because they also showed your pulse rate, your heart rate, which was mainly what we were focusing on. But if you remember, that other number was your percent oxygen saturation. That means how much of the oxygen carrying capacity is actually realized in this red blood cell. And if you remember, most years were 95 or above. That means that of the hemoglobin molecules in here and the heme molecules on them, over 95% of them were in fact carrying an oxygen molecule when you were being evaluated with that little pulse oximeter. You're incredibly efficient. Okay, so yeah. So the importance of this molecule is obvious. You need it to supply oxygen to the tissues. And so thinking about what can go wrong with that, well, we know that these red blood cells lead a difficult life. Now, I have also loaded on Moodle the blood flow through frog webbing, and I'm going to pull that up here quick. Okay. And the idea with this video is to give you the sense that red blood cells lead a tough life. If you look at these blood cells, they have to really squeeze through some tight spots. And over here, we talked about this previously, that red blood cells are actually sometimes flowing through capillaries, which themselves are smaller in diameter than the red blood cells. So that the red blood cells actually have to taco over to fit through that tube. And that's what's happening here. And as they're doing that, they're actually really coming in contact with the side of that capillary wall. And that additional contact means that they have more surface area in contact and do a better job of moving material, oxygen into the cells and getting carbon dioxide out of the cells. They do a better job of that. So that's a good thing. But it also means that they wear out pretty quickly. So these blood cells have a short life, about 100 to 120 days, I think, is what I have in your notes. And they lead such a tough life that after that amount of time, they wear out and have to be recycled. So when they are worn out, they are recycled chiefly in a couple of main areas in the body. The liver and the spleen are kind of main areas where they get recycled. And they get broken down, and your body will reuse what it can. Of that heme molecule, of course, the proteins, that's easy for them to get reused. They just get broken down, the amino acids broken down and reused elsewhere. Some of them get excreted, but most of them get reused. The heme group, the iron gets recycled, but the remainder of that heme molecule gets broken down into two other molecules, bilirubin and biliverdin. Okay? And those two molecules create some rather colorful structures in your body. Now, normally in class at this point, I would ask if any of you have a bruise. Well, I got a picture of a bruise here. That shows up pretty well. Now, the colorfulness of this bruise is created by bilirubin and biliverdin. So when somebody gets hit hard, and what happens is, in the shallower portions of the skin, blood vessels are ruptured, there's bleeding under the skin, subcutaneous, and that blood is represented here by these, not represented, it's creating these purple and red areas. This is actually blood that is leaked out into the tissue. Okay? Okay? Now, the first blood to leak out into the tissue is right here, and this has actually started to break down, and it creates that green color. That's biliverdin. Okay? This yellow-orange color here is bilirubin. It's what's creating those. And so that's what creates a very colorful bruise like that. Again, blood, actual blood leaking out of the tissue, biliverdin and bilirubin. Now, the presence of bilirubin also can indicate diseases. Now, if you remember, the liver and spleen are actively involved with breaking down and recycling the components of blood. If your liver isn't working well, it's not going to do that well. And so you'll get a yellowish-orange hue in the tissues in places where it's not supposed to be. And we call that jaundice. Yellow jaundice is another name for it. And again, here's a picture of a gentleman that is displaying those symptoms. And you can see it really shows up in the sclera of the eye, which should be white. Well, the buildup of bilirubin there has created a very yellow condition. Maybe more commonly, you'll see it in babies. Babies are born, if they're born a little bit prematurely, or if they do have some liver dysfunction, you'll again get bilirubin building up in the tissue. Now, the reason that premature babies, and I'm betting some of you folks probably went through this as children if you ask your parents. If you're a little bit premature, the last organs to develop, the last main organ to develop is the liver in babies. And so if you're born just a little bit premature, your liver isn't fully functioning yet, and so it's not able to take that, to actually process that bilirubin, which it's going to add to bile. Actually, biliverdin is what gives bile its green color. But bilirubin will show up in the tissues. And so they'll take a baby and they'll put it under a light that helps break down that bilirubin until the liver is functioning well. Our oldest, our son, our firstborn, came 11 days early. And so he was a little jaundicey when he came out. And so he came out yellow-orange, and it just so happens that he was born right on Halloween. He came out looking like a pumpkin. Not only that, but in the process, my wife was, what's the word I'm trying to remember? She was breathing too rapidly, hyperventilating. So they told her to purse her lips a little bit. And so she did, but it came out like this. On Halloween with an orange baby. It was just perfect. So anyway, that's what can happen. And in an older gentleman with liver disease, there's another look at what it can look like in the sclera of the eyes. It'll show up in the skin here all over the place as well, but it really shows up very well in the sclera, and it indicates liver disease. Of course, people that consume too much alcohol, this is a common problem that they have. Okay, so that's the breakdown products of blood. And we're talking about how these blood cells work. And so when we do that, oftentimes it's nice to think about how they don't work too, and some ailments that come along with that. Since we're really focused on ailments right now in this country, I thought I'd mention another important one having to do with red blood cells, and that is sickle cell anemia. Now, if you remember, anemia, there are lots of ways to define anemia. So if I just, and I think this is the definition I gave you in class before break. It's an insufficient supply of oxygen to the tissues. Now, as I say, there are lots of ways to define anemia, but however you define it, usually it comes down to some aspect of this, some issue involving this, and an inability to supply sufficient oxygen to the tissues. Okay? There is an ailment that's very common in people of African descent and of Asian Indian descent, and it's called sickle cell anemia. And I'm going to erase this in just a second, so you can always pause this, and if you didn't get that down. That's something you can recover. Oops, that's not the one I want. This is the one I want. Okay, here's a normal red blood cell. Maybe I'll pull this down for the time being. This is a normally shaped red blood cell that would be functioning normally, has all the hemoglobin, supplies the oxygen, does its thing. This is a sickled cell. And if you can remember, now think back to that blood flow through frog webbing, and this sickled cell doesn't go as well through the capillaries, not just because of its shape, but because it also develops a rigidity. It doesn't taco over as well as this would. And so it gets clogged in those capillaries, and it prevents blood flow to the tissues, and you have oxygen-deprived tissues. So if I go back to that video for just a second. Okay. So if you've got a sickled cell, it's going to get stuck right here, and that'll prevent blood flow to the tissues beyond, and you have oxygen-starved tissues. Okay, here's my mouse, there it is. Okay, so there's a very interesting adaptation that's going on here, though. In people of African or Asian Indian descent, roughly, well, it's less than 1/4% of that population in the United States. I only have information from the United States. Less than 1/4% of the population of people of African or Asian Indian descent have 2 genes for this trait, and that means their cells sickle and they have a difficult life. They're anemic. There are all kinds of issues that come along with that, and it's a difficult situation. In that same population, there is slightly more than 8% of that population that has only 1 gene for sickle cell anemia. And in that population, they have normal red blood cells almost all the time. Now, in Africa, Asian India, one of the main maladies is malaria. In fact, malaria is the #3 killer worldwide. Kills that many people, just for kicks and grins. Guessing you're wondering what the #1 killer is. It's diarrhea. Yeah, you say, well, that's a secondary result of another disease. Yeah, that's right, but that's the #1 killer worldwide is diarrhea. And it usually results in dehydration that becomes lethal. Anyway, malaria is a big deal. So, and just to show you what the malaria life cycle looks like, again, this is on Moodle, okay, so you can look at it in more detail. So let's start right here with #1. So you've got a mosquito carrying a parasite, and that's what malaria is. It is a parasitic infection. That mosquito carrying the parasite bites somebody. And whenever a mosquito bites you, they inject a little anticoagulant first. Remember, keeps blood from clotting. They want to do that so they can continue to suck blood out of you. So when they bite a person, they inject stuff into you first. Well, some anticoagulant like heparin goes in first, but also goes into these sporozoites, this parasite. It goes into the person's body. Goes first to the liver cell, infects liver cells, goes through another life cycle. Boom, those liver cells blow open. That other life cycle then finds blood cells, infects red blood cells, and it can take one of 2 paths here, but either path results in a ruptured or destroyed red blood cell, so the person feels very tired, very anemic, very sick because of their loss of blood. Just to complete this, the mosquito then, that parasite will go into another life cycle. The infected person will be bit by another mosquito and will suck up some of that parasite. That parasite will go through this stage in the mosquito. That now infected mosquito will go bite somebody else, and you have the spread of the disease from person to person because of transmission by a mosquito. Here's an interesting thing relative to sickle cell anemia. In a person with 1 gene for sickle cell anemia, now remember, they're going to have just 1 gene, which means they have normal red blood cells almost all the time. But when